Hi, everyone. My name is Professor Gilmore, and today we're going to be talking about some tips on how we can take multiple choice exams as easily as possible. We're going to do this in the context of organic chemistry because that's the only thing I know. And so we'll go through a couple of examples on how we can best attack these types of problems. So when we look at these types of quizzes, exams, whatever they may be, the most important thing is that the answer is given. It's going to be one of those, 4 or 5 options. And so we want to be able to use those answers in order to solve that problem. We don't need to solve that problem on its own and then look to see if that answer is there. We can work back and forth in order to more effectively solve that problem. So the easiest way that we can do this is to first, read through the question, see what kinds of things we're looking for, and then immediately go to the answers that are given. And first, we're going to look at what are the differences in these between these answers. You're going to use these to identify things that are obviously incorrect because they don't have the right number of atoms or whatever it may be. And we're going to try to narrow these things down that way. Next, what we're going to do is to then compare these differences to any kind of unique point in the problem. Does it have a weird group in this position? Have we done this or that? We're going to use that in order to start to immediately eliminate some of those potential answers to make this problem a little bit easier for us to solve. And at that point, once we've narrowed things down a bit, we can then use our knowledge of this area that the question is asking for in order to solve that problem, or at least now we're going to have a much better point to guess as to what this is going to be. So let's look at a couple of examples of what this looks like. First one is solving an NMR problem. We often see this in organic chemistry. And so reading through the problem, we see that we have a proton NMR. We see some peaks down here in red. And we have then the integral values down here in blue, and it's giving us the splitting of these peaks here at 16 hertz. So first up, what we can start to do is to look at any kind of differences or unique features here. And we look at our problems. All of these have some sort of aromatic ring here. On all of these problems, we also have esters that are involved in all of these, and some sort of double bond in there. Although this one now does not have an ester, so there's a nice point of difference. So when we look at we'll start with the aromatic portion because that has protons. We can see in a proton NMR. Here you can refer to your tables if you have them. If you've memorized where these things show up, that's fantastic. But aromatic protons generally show up between kind of 7 and 8 or so. And we see this nice set of peaks here where we have for 2 protons and 3 protons apiece. That is going to be our aromatic protons here. And that means we have 5 aromatic protons. And so now what we can do is we can now go back to our options here and get rid of anything that doesn't have 5 protons on them because we know that those things are simply going to be incorrect. So here we have 1, 2, 3, 4. We know that's wrong. Let's get rid of this so we don't confuse ourselves. Here we have 1, 2, 3, 4, 5. That is still an option, so we'll keep this on the table. 1, 2, 3, 4. That is again wrong. We're going to cross this off. And now we have 1, 2, 3, 4, 5. That's also an option. So just like that, we've already eliminated 2 of these species here. Let's look at some other kind of identifying things in that NMR to make sure everything matches. We have a singlet here for 3 hydrogens. That's going to be a methyl group, and it is between kind of 3.5 to 4 or so. That's typically something that looks like this. Do we have that? Yes, we do. We have that in both of these. We have this methyl ester here and that methyl ester here. And so now the only differences and the only other protons we haven't dealt with yet are, we'll do this in red, we have this double bond and this double bond. And that's going to correspond to these 2 peaks here at that we haven't used yet. They're splitting something called 16 hertz. If you remember what that means, what these coupling constants are, that's great. If you don't, at this point, you can't solve that problem, so you can now guess between these two options, but at least we've narrowed this down to a 50-50 shot. If you remember what these coupling constants are, if we have small coupling constants, maybe 6 to 10 or so, that corresponds to a cis double bond. If we have ones that are then 12 to 18 hertz, that corresponds to a trans double bond, which this fits into that range, and thus option 4 is our winner. So let's look at another example. Here we have a question of stability, so looking at carbocations. So which carbocation is most stable? We care about stability of carbocations because the intermediates, the stability of intermediates matters greatly in looking at how we're going to define our reactivity here. And if we look at this, we have, this one is a tertiary carbocation. We have 3 alkyl groups attached there. Here we have a primary one. Here again, we have a primary one with 2 fluorines on there, and we have now a tertiary one here. Now, the more substituted a carbocation, the more stable it is. We want to be pushing electron density in there because that carbocation is an empty p orbital, does not have enough electrons on that carbon, so we need to add some more in there through something called hyperconjugation. And so the more substituted it is, the better. Hopefully we remember this, and therefore we can already cross off options 2 and 3, and we can now directly compare these two options here. Now, of the 2 of these, we have on the right-hand side is exactly the same. We just have these 2 methyl groups out there. And so let's look at the other half of this molecule to find some differences. Here we have 3 methyl groups that are off to the side. In here we have this double bond. And again, if you remember the difference between these things, and particularly what the significance of this double bond next to this reactive intermediate means, fantastic, we can solve this problem. If you don't, well, at least now we've gotten down to that 50-50 chance again. But here, when we have this allylic carbocation, we now have a conjugated system where we have those electrons can resonate and share between those 2 positions, and we have resonance structures where we can shift that and share that pair of electrons between these positions here, like so. And that is a stabilizing interaction. Anytime that we can draw resonance structures, it's going to lower the energy and stabilize that system. And so we see that option 4 again is our winner. Example 3. So now we're starting to look at some reactivity here. We have to see what is the starting material that was that gave us this reaction here, or that gave us this product under these conditions. Well, let's start to look at our 4 options here, any that are significantly different. So we look at options 1 and 2. They look fairly similar, same number of carbons, same rough positioning here. All we're differing is this double bond and either that exterior-interior position. Option 3, again, same number of carbons, same connectivity. We're just missing that double bond. And option 4 is now a cyclic system, and we are a bit confused by this. It doesn't look the same as these others. This probably is either going to be the answer because it's very unique, or it's our throwaway option here. We don't know of any chemistry really that can just split this bond like that in order to open this ring up. And so we're going to take a stab here. We're going to just get rid of this thing and work our way through here. So now we need to do is to figure out how to do this problem. So we look at the answer again, we see that we have a bromine in that terminal position there and no other double bonds to speak of. We know that when we look at the rest of this, we have light and peroxides. This is a great clue that we're going to be dealing with radical chemistry, and we're going to be doing some sort of formation of radicals and trapping to form our bromine species here. We can functionalize CH bonds, so we don't necessarily have to have functional groups on there, but under these types of conditions, we typically are going to do that now next to this benzylic position here, because that is where we can just form a stable radical and functionalize these. With these specific conditions, HBr and peroxides, we are typically forming a Br radical that is going to drive our chemistry. This is going to add two double bonds to initiate our propagation step within our radical chemistry. So we need a double bond to react with here. Option 3 doesn't have it. And so let's see what we can do here. Options 1 versus options 2, only option 1 has that double bond in that terminal position that would allow that bromine to add to the outside there to give us our product. Option 2 would give us a different substitution pattern here. And so again, we can now narrow this down to option 1. Last example is getting towards multi-step chemistries and multi-step processes. Again, let's now start to do a coupling reaction to form molecule A, and then we react sodium and then ammonia and whatever chemistry that does in order to give us this product. So one of the three main rules for organic chemistry is that negatives attack positives. Most of what we deal with, we have some sort of negative species here. We know these two things are going to react together to give us something. And hopefully we remember that negatives attack positives like so in order to give us this new bond in red. And we have now 3 carbons over there, 1, 2, 3, adding these two together. And so we can immediately start to narrow this down and look at, does all of our answers have the right number of carbons? If we look at option 1, so in our product, we have 1, 2, 3, 4, 5. That comes from this first piece here, 1, 2, 3, 4, 5. And then we have 6, 7, 8 coming from that second group there. Does everything have 8 carbons? 1, 2, 3, 4, 5 in that first one. That is wrong. Option 2, we have 1, 2, 3, 4, 5, 6, 7, 8. That's still on the table. Get rid of this. Option 3, we have 1, 2, 3, 4, 5. That's also wrong because we haven't brought together those two pieces. And option 4, we have 1, 2, 3, 4, 5, 6, 7, 8. So that's still on the table. So now we need to figure out what on earth sodium and ammonia does if you don't remember, or really what is going on. We know that we still have that triple bond here, and we look at our two products, we then either have a cis or trans double bond in that position. So we're doing some sort of hydrogenation here. We may not remember what sodium and ammonia does. Hopefully remember some chemistry of alkynes. So if you remember that this is always going to give us that trans product, fantastic, you've solved the answer. Maybe you don't remember, maybe you remember other chemistry. We know that if we take triple bonds and react this with H2 and poison platinum, some sort of lead in there, something like this, something called Lindlar's catalyst. We know that we can reduce that to the cis double bond. And since this is not this, then we can eliminate this one as well. Or again, we're now back to that 50-50 chance. So when you're taking these multiple choice quizzes, again, the most important thing to remember is that the answer is in there, and we need to use those 4 to 5 answers in order to solve this problem. So slowly work through, eliminate as many as you can, and then you can hopefully solve the problem. Best of luck and see you later. All right, bye.